Dear audience, my name is Tom Hensinger, and I'm the president of ÖAW-IST Austria, and it's my great pleasure to welcome you to today's ÖAW-IST lecture. The Austrian Academy of Sciences and the Institute of Science and Technology Austria are the two largest non-university research organizations in Austria. While the former is more than 15 times older, I should say, than the latter, our two institutions have an overlapping interest in many fields, from physics to mathematics to biology. And it was 4 years ago that we got together and decided to inaugurate a new annual lecture series, the ÖAW-IST lectures. The common aim of this series was and is to bring to Austria speakers of the highest international standing, speakers in the fields in which we have common interest, and speakers that are of interest to a wide public. In the last years, we had been able to welcome Nobel laureates David Gross and Christiane Nüsslein-Volhard, also Fields medalist Martin Heira, and computer scientist Bernhard Schöllkopf. And today we are continuing the series with an equally eminent speaker, but for the first time not in the lecture hall of IST nor in the great auditorium of the ÖAW, but online. And we are happy to see wide interest in this format also. I wish you an interesting hour, and I may now ask the president of the ÖAW, Anton Zeilinger, to introduce today's speaker.
Hi to everybody from my side. I see that the number of people watching this is increasing. I hope it increases exponentially, but we'll see how it goes. I would like to warmly welcome Angelika Amon. First, a few, she's among many other things, she, as is Tom Hensinger, is a member of the Austrian Academy of Sciences. And therefore, I would like to say a few words about the Academy. I wasn't aware that we are 15 times older than IST. At some time, it must have been nearly infinity, but this factor is certainly going down in the future. Just a brief word, like most academies in the world, this academy was created rather late. This has specific reasons, but as a consequence of the freedom of ideas following the Age of Enlightenment. We were created in 1847, and a specific nearly unique feature for an academy in the West is that we run research institutes. We run about 29 institutes from all fields. So some fields have been mentioned by Tom, where we have overlap, but we also have fields in the humanities, and it's quite interesting. We have, for example, some of the top experts who know how to talk to local people if there is a problem in the Near East, like a terrorist problem and so on. They have always historic knowledge, lots of music research, etc., etc. So it's very interdisciplinary, and that makes it quite fascinating. So I would now like to introduce our speaker today, Angelika Amon. She went to the same high school. Here we saw gymnasium as I did. I mean, not quite. There are two buildings, a little bit separated, but at that time one was the boys' school, the other one was the girls' school. We never met in school because Angelika was born after I graduated from my school, but it would have been fun actually. Anyway, Angelika received her Bachelor of Science and her PhD from the University of Vienna. and then followed a beautiful international career. I mentioned a few points. The postdoc fellowship with Ruth Lehman at the Whitehead Institute. Then she became a Whitehead Fellow, Whitehead Institute at MIT. Then since '99, she's at the Koch Institute at MIT for Integrative Cancer Research, and Kathleen and Curtis Marble Chair for Cancer Research. Many honors. They include the Alan T. Waterman Award in 2003, the National Academy of Sciences, US National Academy of Sciences Molecular Biology Award, the Ernst Young Prize for Medicine, and probably one of the most significant ones, the 2018 Breakthrough Prize in Life Sciences, 2019 Wilczek Prize in Biological Sciences. Among memberships, the Howard Hughes Medical Institute, the National Academy of Sciences, the US National Academy, our academy here in Austria, Foreign Associate at EMBO, etc. And she's also a member of the Scientific Board of IST Austria. I know this at latest, since we wanted to have you as a member of our foreign board, but we have a rule that people who are at other institutions in Austria should not be on our boards, but maybe someday we can ask you again.
I would be delighted.
Would be nice, yeah. Would be nice. Concerning the research topics, I can only read here what they wrote down for me, because I'm not a specialist. This includes molecular mechanisms that control cell growth and division, and also studies on how errors in this process lead to diseases such as cancer and the Down syndrome, and how they impact the aging process. Actually, a question which becomes for everyone more and more important the older we become, personally. So without much further ado, I would like you to start with your talk. The title is Why Big is Bad, Decrease in DNA Cytoplasm Ratio is a Cause for Senescence. I understand that you guys, the broad audience, can ask questions. You can submit written questions during and right after the talk. There is a question and answer feature of Zoom, which you can use. And Angelika will then later read out the questions and answer them after the talks. So please, Angelika, go ahead.
Thank you very much, Tom. Thank you very much, Anton. It's a real pleasure to give this lecture. I live far from Austria, but I will always be an Austrian at heart, and so it's always a very special honor for me to be able to have the opportunity to give a talk back in Austria. I wish I was there in person, but unfortunately the circumstances don't allow it. And I still hope that I'll be able to convene sort of a new idea that we have, that basically the ratio between the amount of DNA and cytoplasm a cell has is really a key determinant of the fitness of a cell. And we propose that as this, over the lifetime of a cell, this ratio changes, and we propose therefore that this change is a cause of senescence. So before I start, I want to introduce to you the two people who've done this work. Two very talented postdoctoral fellows. The work was initially started by Gabriel Neurer, who is now at the ETH, he's now at the, as an independent position at ETH Zurich, and Jette Lengefeld, who is currently a postdoc in my lab. Before I want to start out and tell you about our science, what I wanted to sort of you guys keep in mind throughout my talk is two things. that I think the work that I'm going to tell you about actually will also shed some light on these very, very basic facts about cells. So stem cells are cells that give rise to our tissues and that renew our tissues. And something that I always thought was very peculiar about stem cells is that they're exceedingly small. I'm giving you some examples here. So hemopoetic stem cells are the stem cells that give rise to the blood cells. And you can see their size is small, very small compared to the differentiated cells, the cells that they will give rise to. We see that in adipose tissue. Adipose stem cells are tiny compared to the fat cells in our body, mesenchymal cells, and even embryonic stem cells, human embryonic stem cells are tiny. They're really the size of yeast cells. And what I'm showing you here is a very extreme example of sort of the difference between stem cells and the differentiated cells that they give rise to, and this is the muscle cells. What you see here is this is only a very small part of a muscle cell that is a very long syncytial cell that is huge. It's just many cells come together and fuse. And what you see here in blue is what's called a satellite cell. That's the muscle stem cell. It is tiny compared to the cell it will give rise to. The second fact about cells that I would like you to keep in mind is that cells come in many sizes and shapes, and I'm showing you two very extreme examples here, an oocyte and sperm. And you know, these oocytes are huge, the sperm are unbelievably tiny. But what's important to note is that even though the size of cells can vary greatly between cell types, that within a given cell type, the sizes are very narrowly distributed. And that, of course, raises the possibility that raises the question of whether a certain cell type is important for cell function. And this is the first question. This is one of the three questions that I would like to address today. I would like to ask, is a cell type specific cell size important? And then in what follows is sort of, what if we change that cell size? Will that change the properties of the cells in specific ways? And then finally, I would like to discuss the possibility that a cell size increase without the corresponding increase in DNA content, could that be a cause of aging? So before I start, I want to tell you something. There's one important fact that you need to know about cells that is actually, I find, significantly underappreciated. But it's true for all eukaryotic cells. And that is that cell growth and cell division are only loosely coupled. So what does that mean? Cell growth is the accumulation of biomass when a cell grows in size, when it gets bigger. Cell division is the process by which you make two cells from one cell. And people already realized in the 1970s that when cells arrest in the cell cycle, because, for example, they got damaged and they need to stop dividing to repair that DNA before they can continue, they will increase in size. And they will do so in the absence of a corresponding increase in DNA content, because obviously their cell cycle is stopped, so they can't make more DNA. And I want to show you this example here for budding yeast. What I'm showing you here is that there's a budding yeast cell that divides, and then we use either genetic manipulations or chemical manipulations to block these cells from growing. And so this cell gets stuck in the cell cycle. This is a G1 cell. And as it gets stuck, in this particular case for 2 hours, it starts growing. But then as you can see, it's been stuck in this cell cycle stage for 6 hours. These cells become dramatically large. And so that indicates that cell growth, to some extent, is independent of cell division. And this is a very important fact that really I think provides the foundation for what I'm going to tell you about. So let me now start by telling you how we got into this, into addressing the importance of cell size for cell fitness and senescence. So Hayflick and Moorhead in the 1960s described for the first time senescent cells, which were basically cells that could no longer divide. And what they noticed in their very first publication is that these senescent cells became very, very large. And in fact, they called them pancake cells. They were so large. And this very simple observation leads to a very simple question, which is, could large size be a cause of cellular senescence? And we decided to address this question, and the way we decided to go about this was to simply ask, well, if we take a young cell and we make it big, will this cell then have features of senescence? And so we first did this work in budding yeast, and then subsequently we also addressed this question in mammalian cells. And I'm going to first start telling you about our work in budding yeast. So Alexei Goranov, when he was a postdoc in my lab, developed a system to basically change the size of a cell without changing its DNA content. And he took advantage of mutants in budding yeast that are defective in cell division. But they're not generally defective in cell division. They're what we call conditional mutants or temperature sensitive mutants. What that means is that the protein works at the permissive temperature, usually room temperature 25 degrees, but it no longer works at a higher temperature of 37 degrees. And so what you can do is you can take these cells that are proliferating at 25 degrees, and then you can shift them to the higher temperature, the restrictive temperature at 37 degrees. And what will happen is that these cells will get stuck in the cell cycle so that DNA content can no longer change, and they will slowly increase in size. So basically what we're doing is with time, we are basically decreasing the DNA to cytoplasm ratio in these cells. And so I'm showing you this example here again, where we are resting these cells. So after 2 hours, they are still a reasonable size, but then after 6 hours, they get very, very large. And what we can then do is we can then remove the cell cycle block and then can then compare the properties of these cells that didn't grow as large during the arrest with the properties of the cells that grew very large during this arrest. Of course, comparing cells that were arrested for 2 hours with cells that were arrested for 6 hours is not a great experiment, because God knows what happens in those 4 hours. Many other things unrelated to cell size could have started affecting the cells in some way. So an additional control that we're introducing is shown here, is where we keep cells for the same 6 hours at 37 degrees, but at the same time, we're preventing these cells from growing by, for example, in this particular instant, treating them with a chemical that inhibits protein synthesis, cyclohexamide. And so what we can now do is we can compare the properties of cells that have grown really large during the 6 hour rest with the properties of cells that have also been stuck at 37 degrees for 6 hours, but that remain small. And differences in the behavior of these two cell types will then basically allow you to make conclusions about how cell size or a decrease in DNA cytoplasm ratio affects cells. So Gabriel did this experiment, and he can show very nicely. What we're looking here is we're looking at cell volume over the time of the arrest. And we would just take this one mutant. As you can see, it grows larger over time. And then we have various manipulations, protein synthesis inhibitor, or simply starving the cells during the arrest. And what he finds is that these cells do not grow as large. And what we can then do is, so then we have populations of cells that have grown really large, and populations of cells that have remained small during the arrest. So this is the manipulation we did. We can then remove the cell cycle blocks and the various drugs and sort of allow the cells to resume to proliferate and ask the very simple questions. Well, how do the large cells do, and how do the cells do that stayed small during the arrest? And this is a very simple experiment. We're basically assessing the growth properties or the proliferation properties of these cells. So we arrest these cells for 6 hours, they get very large, and then we simply drop them on a plate and ask them, well, how quickly can they grow up? And so when they have not been grown large, they grow up very well. That you can see there's a lot of highly dense cells growing up here. But when we grew them large for 6 hours and then asked them to resume proliferation, you can very clearly see they have great trouble. They're producing a lot fewer cells and they're growing much more slowly. What was interesting is when we kept the cells small during the cell cycle arrest by cyclohexamide treatment or by starving them for glucose, these cells resumed proliferation just fine. And so then what Gabriel did is, I'm not going to show you the data because it's published. What we found is that when we looked of why these cells have trouble proliferating, what we noticed is that every cell cycle stage was affected. Nothing really was working in these cells anymore, indicating that there was a very systematic and almost systemic breakdown of proliferative abilities in these large cells. And that then, of course, led Gabriel to ask, well, if proliferation is so profoundly affected, could it be that other cellular processes, such as induction of transcription, cell signaling, and whatnot, is also affected by large size? And the answer was that yes, every cellular process that he looked at was actually dramatically affected when we changed the DNA to cytoplasm ratio. And I'm showing you one example here, which I believe is quite a dramatic example. What I'm showing you here is I'm showing you a promoter, an inducible promoter that's hooked up to the expression of a green fluorescent protein. And basically we're inducing this promoter in normal cells that are still small, that have only been arrested for 2 hours, in cells that are large, that have been arrested for 6 hours, or in cells that have been arrested for a long time, but that have been kept small during the arrest. And what I think is very dramatic is whereas the small cells are very able and very quickly able to induce this reporter construct upon induction of the promoter, the large cells really failed to do this at all. And so then Gabriel continued to conduct a very systematic analysis. And as I mentioned before, every process that he looked at, cell proliferation, cell signaling, gene induction, were affected by these large cells. And so the fact that every process was affected sort of let us step back and ask, clearly there's something very profoundly wrong with these large cells. And maybe we should just make some very simple measurements of RNA, protein, and other macromolecule in these cells to ask what is going on. And when Gabriel did that, he really made a very surprising discovery. What I'm showing you here on the left is where we're measuring very simply the volume of these cells over time. And I showed you this before, these cells get larger. And at the same time, then he looked also at total protein and total RNA. What he observed is that while initially as these cells were growing, those three parameters were scaling, meaning they increased coordinately. As the arrest continued, basically RNA and protein synthesis stopped scaling with volume. And obviously as a result, as macromolecules decline in the cell, the density of the cell also decreases. And that's shown here. These are individual cell density measurements where we're looking over cell volume over time. and correlating that with cell density. And as you can see, over time, as these cells grow large, cell density declines. When we further asked, well, what actually happens to the protein composition of these cells as they grow larger, we used total cellular proteomics to assess this. What we find is that especially factors involved in transcription and translation dramatically drop as these cells grow larger. And that's shown here. So what we're comparing here is the protein composition 3 hours into the arrest, right here. So at that point, cell volume and macromolecule synthesis still scale. With a 5-hour rest, where that scaling stopped, what you can very clearly see is that proteins involved in transcription and translation become downregulated in these large cells, and stress proteins become upregulated. And consistent with this downregulation of transcription and translation machinery is when we then start looking at growth rate, meaning the ability of cells to make macromolecules over time. What we see is that initially during the time course, we see a steady increase in growth rate that correlates with cell volume increase. But then as these cells become large, the scaling breaks down. So what does that mean for the cell? Basically what this means is a very simple thing, but that obviously has very dramatic consequences. So as these cells arrest for long periods of time, basically what happens is that the cytoplasm becomes diluted. And as I'm sure you all remember from Biochemistry 101, once you start diluting extracts, every enzymatic, every chemical reaction will basically stop working. And so the key question then, of course, became is sort of why does at some point during this growth period macromolecule biosynthesis stop scaling with cell size increase? And a very simple hypothesis that we had is that probably, something that could happen is remember, as these cells grow in size, we're not increasing their DNA content. And so Gabriel asked a very simple question, well, could it be that the DNA becomes limiting? And budding yeast is a really ideal system to address this question, because budding yeast cells come in two flavors. They come as haploid cells, and they come as diploid cells. And importantly, as we've known for forever, is that a diploid cell is twice as large as a haploid cell. And so what we can do now is we can take these haploid cells and diploid cells, and we can grow them to the same size and ask all these cellular processes that break down in large cells, are they less likely to break down in diploid cells that have twice as much DNA as in haploid cells? And we've done this with all the analysis. I'm just going to show you one example here. Recall, I showed you this promoter that loses induction as these cells get larger. And what you can very clearly see, when the haploid cell is still small and of the proper size of about 50 femtoliter, we can induce this promoter quite easily. As these cells grow at 150 femtoliters, this ability drops and then is completely lost at about 270 femtoliters. Importantly, what we find is that when we do the same experiment in diploid cells, what we see is that the scaling breaks down at exactly twice the size. So at 150 femtoliters, the diploid cell that has twice as much DNA as the haploid cell is still capable of inducing this promoter quite readily. At 270, it stops working so well, and at 550, it doesn't work at all. So from these experiments, we basically concluded that when cells exceed a critical size and become extremely large, that the DNA becomes limiting in cells. So you say, okay, this is interesting. You made these very large cells. That is a sort of a very highly artificial situation. Why should I care about this? And the question that you should ask is, when do yeast cells ever reach such large sizes? And it turns out that during the normal physiological life cycle of budding yeast cells, it never reaches sizes where this starts happening. However, as budding yeast cells age, they do become much larger in size. And that has been known for a long time, since Mortimer first described that budding yeast cells can only produce a finite number of offspring, that these very large cells, these very old cells, excuse me, are actually indeed quite a bit larger than normal young cells, as you can see here. And so this observation then, of course, begs the question of that perhaps one reason why these aged and old cells lose their proliferative abilities is because they have experienced a significant decrease in DNA to cytoplasm ratio. And so a simple way to address whether that is the case is to basically take a young cell that should be able to produce a lot of offspring and make it fat, and then ask, does it have a shorter lifespan? So we return to the experimental system that we developed to make these cells, to increase the size of cells in the first place. So I'm showing you here, this is basically an aging yeast experiment where we're assessing how many divisions a yeast cell can, how many offspring a yeast cell can produce over its lifetime. And as you can see, a cell that's of normal size, that's been arrested in G1 for about 2 hours, has an ability of producing a maximum number of about between 20 and 25 offspring. When we now make these cells really large by arresting them in G1 for 6 hours and they get really big, what you can see is that their ability to produce offspring dramatically decreases. However, when we keep the cells small during that arrest by either treating them with a protein synthesis inhibitor or by starving them, you can see that we at least partially suppress this loss, this decreased ability to produce offspring. So from these and many other experiments that I'm not going to show you today because they are published, is we concluded that a large size could contribute to aging phenotypes in yeast. So that was all nice. I mean, so we probably found one reason of why budding yeast cells age. They become really large, and that leads to a decrease in DNA to cytoplasm ratio, and that's, you know, makes biological processes from working well. But we really wanted to sort of ask whether this was true not just for budding yeast, but for eukaryotes in general. And so we turned to the mammalian system and basically repeated the same experiment that we did in budding yeast. So we took mammalian cells, and rather than using mutants to stop the cell cycle, we used a drug. In this particular experiment, we used a drug that's called palbociclib, or short PD, that arrests cells in G1. And like in budding yeast, what you will see is that when you arrest these cells in G1, they get stuck in the cell cycle and they will increase in size, importantly, without changing their DNA content. And what this graph shows you is that that works, right? So we can arrest these cells with PD and they get quite a bit larger. So will that impact the fitness or the proliferative abilities of these cells? And so to address this, then what Gabriel did is he washed out the drug and then asked whether these large cells can resume cell proliferation again easily. Oh, I forgot. I should have showed you that when we starved the cells during that palbociclib treatment, during that arrest them in G1, we can suppress this increase in cell volume, which is obviously always the critical control that we're using in our experiments. And this experiment is shown here. So we are assessing the ability to resume proliferation after these cells have grown large. And as you can see, these very large cells that were treated for long periods of time with this CDK4/6 inhibitor, they have great difficulties in resuming proliferation. However, when we starve the cells during the PD treatment and keep them small, their ability to resume proliferation is partially rescued. So these experiments suggest that indeed, when we culture mammalian cells in a dish, we can mimic senescence, because basically the inability to resume proliferation is a key characteristic of senescence. We can mimic senescence simply by growing cells large. That was interesting, but this was an in vitro experiment of cells in a dish. And really the key question that we wanted to address is, does this have any relevance for mammalian cell biology in vivo? And so Jette Langefeld decided to take on this question, and she adapted a truly marvelous system to very carefully, that allows you to very carefully examine the fitness of cells in an animal in vivo. And this is through hemopoetic reconstitution experiments. What's so beautiful about these hemopoetic reconstitution experiments is two things. So first of all, when you lethally irradiate a mouse, it will destroy its bone marrow and therefore its ability to make blood, and these animals will die. But we can rescue, or we can prevent these animals from dying by giving it hemopoetic stem cells, the stem cells that give rise to the blood lineages, from a donor animal. And what's really neat about this is that we can take these HSCs, so short for hemopoetic stem cells, we can take these HSCs, we can either genetically or chemically manipulate them before we inject them into a lethally irradiated recipient, and then ask how these manipulations affect the fitness of these hemopoetic stem cells. The second very powerful aspect of these hemopoetic reconstitution experiments is that hemopoetic cells come in two flavors, meaning they have a surface marker called CD45 that comes in two types of alleles, CD45-1 and CD45-2, and they can be very simply distinguished by very simple staining. So what we can do is we can take donor HSCs, inject them into a lethally irradiated mouse that has a different CD45 surface marker than the donor HSCs. And then by simply analyzing how many blood cells of the CD45-1 versus CD45-2 flavor are in the blood of this recipient animal, we can very carefully and very sensitively measure the fitness of these HSCs over time by simply analyzing what the donor white blood cell contribution is to the animal. And so Jette decided to start out again with a very simple experiment. What she decided to do is she decided to isolate hemopoetic stem cells from a mouse and sort them by their size. And she developed a sorting method that allowed her to isolate what she calls X small, medium, and X large HSCs. And these cell volume measurements show you that she's indeed able to isolate stem cells of different sizes. What I want you to notice is that these differences are actually not that big. It's only about 30%. But then what she can do is she can take these stem cells of various different sizes and inject them into lethally irradiated recipient, and then monitor the ability of these differently sized HSCs to contribute to the recipient's blood over time. And what she found was something quite dramatic. Whereas the small and the regularly sized HSCs were very able to contribute to the blood lineage of the recipient animal, the X-large HSCs were completely defective in their reconstitution potential, indicating that these cells are profoundly unfit in some way or another. So Jette then, of course, wanted to go on and ask, well, why can't they reconstitute the hemopoietic system of a recipient animal? And what she did is basically during these reconstitutions, what has to happen is that the HSCs they get injected into the bloodstream, but they actually have to go to the bone marrow niche to reestablish the hemopoietic system in a process called homing. What she found is that size did not affect the ability of stem cells to home to the bone marrow niche. It also didn't affect the ability of these HSCs to differentiate into the various different hemopoietic lineages, but it had a very dramatic effect on the ability of these HSCs to proliferate. So these results indicated that naturally large HSCs have a decreased ability to proliferate and repopulate the hemopoietic compartment of a lethally irradiated recipient. Can we artificially change the size of HSCs and see the same effect? And again, Jette went back to the system that we used before in vitro, where she's treated mice with this drug, palbociclib, the CDK4/6 inhibitor, for prolonged periods of time. And then she was indeed able, by injecting these animals for a long time, to increase the size of these HSCs. We found that about a 20% increase again in size. It's a subtle increase in size, but nevertheless very significant. And again, now this is the control you're very familiar with. Obviously, we want to avoid, this drug could do 1,000,001 things to these HSCs. So we really wanted to make sure that the effects that we're looking at are due to changes in size. So what we did is at the same time, we treated cells with a drug that would reduce macromolecule biosynthesis, and therefore during the PD treatment would prevent these HSCs from getting large, which is indeed what we can do. And again, the result that Jette saw was quite dramatic. The cells that were, the HSCs that were large because the donors were treated with PD, basically completely failed to reconstitute the hemopoietic compartment of a recipient animal. But HSCs that were kept small during the PD treatment by also treating them with rapamycin were quite capable of reconstituting the sickness. So these results were interesting. They suggested that indeed cell size seems to be affecting the ability or the fitness of these hemopoietic stem cells. And so if cell size was indeed a determinant of stem cell fitness, a prediction of that hypothesis is that conditions that are known to cause an increase in stem cell exhaustion should also lead to an increase in HSC size. And yet it decided to test this idea by looking at three conditions that people had previously described to cause stem cell exhaustion. And one of them was irradiation, serial pregnancy, and aging. And what Jette found was that indeed, HSCs that had been irradiated or HSCs from animals that were serially bred or that were simply old, these HSCs were larger. Again, the difference weren't that great, were small, but the effects on the biology were quite dramatic. So these, of course, are all correlations, and those are nice correlations, but in order to establish causality, really what you want to do is you want to prevent HSCs from getting large during these conditions that are known to cause stem cell exhaustion, and then ask whether if you kept these HSCs small during these various manipulations, do they retain their stem cell ability? Do you protect these HSCs from exhaustion? And so Jette did that too. These experiments are showing here. To me, I have to tell you, this is one of the most remarkable results that I think my lab has ever produced. So what Jette did here is she took an animal and she treated it with rapamycin. So rapamycin inhibits macromolecule biosynthesis and therefore will keep, prevent cells from growing large. She treated them for 2 weeks. Then she irradiated these mice with 3 gray, which will enlarge them normally. And then she would afterwards treat them for another 2 weeks with rapamycin. Then she would isolate these stem cells and assess their ability, their fitness by injecting them into lethally irradiated recipients. And I find this result quite remarkable. So when you irradiate HSCs, they lose their ability to reconstitute a lethally irradiated recipient. But by simply treating these mice with rapamycin 2 weeks before and 2 weeks after the radiation, you basically protect these animals, these HSCs, from radiation-induced exhaustion. So basically, rapamycin is radioprotective. Yet they also asked whether serial transplantation, which is another challenge during which HSCs become very large and become exhausted, whether rapamycin treatment would also protect animals from exhaustion, HSCs from exhaustion during serial transplantation. So what she did is she isolated HSCs, reconstituted recipients, and then treated these mice with rapamycin, so preventing them from getting large, and then isolated these HSCs again and injected them into another recipient. Again, what you can see, rapamycin really protected HSCs from exhaustion during serial transplantation. And the same was true during aging. So when we take young animals and then over their lifetime simply treat them with rapamycin, the first thing that we found is that the ability of HSCs to form colonies, the ability to proliferate, really was greatly preserved when they had been treated with rapamycin throughout their lifetime. And also, their ability to reconstitute a lethally irradiated recipients. So old HSCs, that's been known for a long time, old HSCs have a very poor ability to reconstitute lethally irradiated recipients. But when these animals had been treated with rapamycin throughout their lifetime, they basically, these hemopoetic stem cells retained their reconstitution ability, were protected from exhaustion, and contributed quite nicely to the bone marrow. So from these experiments, Jette concluded that HSCs enlarge under conditions known to cause stem cell exhaustion. And importantly, when we prevent this enlargement under these conditions that induce exhaustions, it protects these animals from stem cell protection. And I'm sure you've heard a lot of popular science that rapamycin and could be the fountain of youth and protect people from getting old. Our data would indicate that if you start taking rapamycin late in life, when your stem cells are already large, that this will likely have no benefit. However, if you had a magical tool that could take these large HSCs and shrink them back to a smaller size, that would have significant benefits. The only problem is that we really don't know of any natural mechanism that allows large cells to become small again. However, genetically, we can make large cells small again, and we can do that by shortening their G1 phases. So there are various proteins that control the G1S phase transition. And when you inactivate inhibitors of the G1S phase transition, these cells will rush through G1 faster and will therefore have a shortened G1 and can become smaller. And the tool that we use is the very famous tumor suppressor gene, the retinoblastoma gene. Deletion of the retinoblastoma gene has been shown to cause cells to become smaller again. And so I'm showing you here what I think is quite a dramatic experiment. So Jette took a mouse that had a conditional allele of this RB gene that we could inactivate at will. And what Jette did is she took this mouse, she either isolated small HSCs or large HSCs. She reconstituted them into a lethally irradiated recipient, then waited for 2 weeks for the HSCs to find the bone marrow niche and get established there. And then by using a genetic trick, excised the RB gene. And so the first key question is, would that actually really make HSCs smaller? The answer to that is yes, that's shown here. So when she took the X-small HSCs, and then excised the RB gene, they did not become smaller, which I actually think is quite interesting because it would indicate that there's sort of a lower limit of size below which you cannot go. But the large HSCs that she isolated when she excised the RB gene, these HSCs became indeed smaller. And what was really quite dramatic was that the large HSCs, I've shown you this before, they really have a poor ability to reconstitute a lethally irradiated recipient. But when we inactivate the RB gene, they now have much greater potential of reconstituting the hemopoetic compartment of these cells than before. So I think these results lead to, I believe, important conclusions. I think what we found is that decreasing the DNA to cytoplasm ratio has wide-ranging effects on cell physiology due to DNA becoming limiting. And our data would further suggest that senescence in mammals may be in part caused by a decrease in DNA to cytoplasm ratio. And I think what our data also show is that very small changes in HSC size can have very dramatic effects on stem cell potential. So I want to put this now into context for you and sort of propose a model for how we believe our data could really provide a universal explanation for senescence in eukaryotes. So as a cell divides over its lifetime, it will encounter damage, foremost DNA damage during DNA replication. So this stochastic stellar damage causes transient cell cycle arrest. You know, if your DNA is damaged, you cannot proceed in the cell cycle. You have to stop in the cell cycle to repair that damage before you can proceed. And what will, of course, happen is during these transient cell cycle arrests, the cells, as I've shown you, will continue to grow. And so now this cell is larger than it was before because it had to stop during the cell cycle and to repair some sort of damage, most likely damage. And so this cell will now grow at a larger size. And what's really important is I told you we don't know of a single mechanism whereby cells can shrink back to their old size once they have grown beyond a critical size. And with number of cell divisions that a cell undergoes as it grows older and larger, obviously the probability of encountering damage increases. So with every damage or every problem a cell encounters during its lifetime, it will get larger and larger and larger. And once the cell will become so large, it will be senescent. And as I've shown you, these large cells then experience all the characteristics that are of senescence, which is cell proliferation defects, cell signaling defects, gene expression defects, and so forth. And I've also shown you this is because DNA becomes limiting. So at this point, I want to circle back to an initial question that I sort of, initial fact that I brought up at the very beginning of my talk, where I said, well, isn't it peculiar that stem cells are really small? Really, the job of stem cells is to proliferate, to engage in cell signaling with its niche, and it has to be very versatile. And so maybe I've shown you that for these processes to work properly, you really need to have a very high DNA to cytoplasm ratio. And we would like to propose that perhaps one reason why stem cells are so small is that to maintain this favorable DNA cytoplasm ratio, to execute these processes that stem cells need to do throughout the life of the organism very accurately. And I think the last question that I sort of want to pose here is sort of a little bit philosophical. It's like, how does too little DNA content cause all these phenotypes? Obviously, there's many right answers by, chemical reactions, enzymatic reactions stop working when the cytoplasm becomes too diluted. But I still think it's noteworthy that most of the processes that are especially affected by large size are processes that rely on unstable proteins, cell proliferation, cell signaling, and transcription. And sort of my favorite idea is that when protein synthesis rates decline due to the DNA becoming limiting, unstable proteins are preferentially lost. And I'm showing you the model here, where we're modeling this to sort of over time, cell protein synthesis rates decline because the cells keep growing and the DNA becomes limiting. What I'm modeling here is the relative concentrations of proteins with different half-lives. And as you can see, the relative concentration of the very short-lived proteins drops much faster than of the long-lived proteins. And so for the people who are not as versed in the Leibniz method, we've made a little movie that sort of illustrates this. So what I'm showing you here is I'm having a bag of blue and yellow circles. We're filling this bag with blue and yellow circles at the same rate, so that these balls get dropped in at the same rate. But the yellow circles have a very short half-life, versus the blue circles have a very long half-life. And what I hope you appreciate is that as you're throwing balls into this bag over time, you really start, the blue circles take over and the yellow circles decline. The concentration of the yellow circles declines. And so sort of give you a little bit of a sense of where we're going to head with this. I mean, given that cell cycle arrest is a very frequent occurrence during the lifetime of a cell, I mean, maybe there are mechanisms in place where cells are trying to prevent excessive growth during cell cycle arrest. Basically, they have their own anti-senescence mechanisms to protect them from getting too large. And we began to investigate this question. We indeed find that there is a response to cell cycle arrest, where cells immediately basically dial down ribosome protein and expression of ribosome protein and ribosome biogenesis genes. And how that's going to work is obviously still something we're trying to figure out, but perhaps in a few years down the road, I can tell you about the answer. And with that, I want to end by again thanking the lab and the many collaborators that we had on this project. This was a great collaboration with Omar Yilmaz, Scott Manalis, Jackie Lees, and Lori Boyer. And here are all the talented people in my lab who did all this work. And with that, I want to thank you for your attention. I'd be happy to take questions. Am I supposed to do something, or is just everybody happy?
You should.
Angelika, I thought, first of all, thank you very much for your great talk. I thought, I was told you would be taking questions yourself on Q&A. I can, of course, read them, but there's the button. The
No,
Q&A
I can do it right away. Sorry. Yes, Q&A. I have a few questions. So, Franz Klein, my good friend, asked me the question of how do oocytes manage? How do oocytes manage? Is the key to quickly shrink during the first divisions of the zygote? Have you increased, decreased RDNA repeats? So, Franz, thank you for the question. Obviously, as you know, oocytes get stuffed with all these machinery to make proteins and protein synthesis. And what we believe is going on is that machinery that sort of gets stored in the oocyte basically gets you over the hump of the first few divisions where you clearly have a dramatically unfavorable DNA to cytoplasm ratio. So you're stuffed with all these goodies that help you produce proteins and other macromolecules, because I'm sure, as you know, in the first few zygotic divisions, there's no involvement of the DNA at all. Transcription only becomes important it depends on which organism you're looking at, but at a very special transition where transcription starts happening. The second question is, how does your work from Yvonne Vallis, how does your work fit in with telomere shortening? I think that's a great question. I think, excuse me, telomere shortening is one of this DNA damage insults that will cause cell cycle arrest. That has to be repaired before cells can proceed during proliferation. So in a sense, telomere shortening in our view is one of the damages, one of the insults that will precipitate the cell cycle arrest that I have spoken about. So as you know, telomerase is not very well expressed in terminally differentiated cells. So at some point over divisions, you will start running out. And that will lead to unrepaired or eroded telomeres. And that will just cause a genuine DNA damage checkpoint arrest that will then cause cell cycle arrest that will cause cells to become larger. Next question, Renato Arnese. Does rapamycin treatment also impact cell cycle and cell division? Is the experiment where mice were treated continuously with rapamycin? Could HSCs have sustained less cell division, thus be less damaged, being then able to better reconstitute? Yes. So to our knowledge, rapamycin does not affect the cell division cycle per se, but of course, it will reduce the ability of cells to enter the cell cycle. But I want you to remember is that what we're doing is we're actually irradiating these HSCs, right? So they will not proliferate at all anyway, because they will be damaged and they will be G1 arrested for these cells to repair their DNA. So during the radiation, which causes significant damage, there is no proliferation to speak of. So I think your concern is a very valid concern, but I don't think it applies here because these HSCs are not dividing following irradiation. Hypothetically, would it possible to rejuvenate cells by adding exogenous DNA increasing? Yes, I mean, I think our budding yeast experiment, where we look at diploids versus haploids, is if you increase the DNA content, that would actually, would certainly rescue these fat old cells, absolutely. So we have thought about the idea of injecting nuclei into these very large cells and see whether we can get them to divide again. You know, there's not a lot of people who know how to do these experiments anymore, so we haven't really pursued it. We've actually also pursued to cut away cytoplasm from these cells and make them smaller again, but that failed miserably for a lot of technical reasons. Joao Matos, how does the increase in cell size caused by CD17 activation compare to the increase naturally seen during aging? Do cells during aging really reach the size? So the answer to your question is, so that the CDC28 mutants that we're using are obviously extreme examples, right? They are growing to 750 femtoliters. And those are not usually sizes that are seen in aging cells. Having said these aging cells usually, you know, they reach sizes of about 400 femtoliters. And when we are looking at scaling between cell volume and macromolecule synthesis, already at about 250 femtoliters, we start seeing that the scaling stops. So obviously, the aging phenotype is not nearly as extreme as the phenotypes that we see in the CDC28 mutants. Basically, the reason why we picked the CDC28 mutants, that is by exaggerating, you know, these changes in DNA to cytoplasm ratio, we were hoping to see phenotypes more clearly. The effects in the aging cells are not nearly as dramatic as in the CDC28 mutants. Leonid Sazonov is asking me what happens to the mitochondria after G1 arrest. I cannot tell you, we have not looked. I mean, maybe I should stop, but maybe I'll take one last question. I don't, please tell me what you guys want me to do. So Doris Chen is asking, what about the size of cancer stem cells? Is it smaller than aged healthy stem cells? So cancer stem cells are usually considered to be tiny, yes. They have an extremely high DNA to cytoplasm ratio. They are basically a nucleus with a sliver of cytoplasm around it. All right.
Maybe if you take a couple more questions, I mean,
yeah, I was.
We have time, but on the other hand, also at some point we have to stop. Right?
Yeah, of course. Florian Kabinger, I have a question regarding the yeast system. Was a transcriptional inhibitor used in comparison to cyclohexamide? If yes, did you see the same effects? So we haven't done this experiment with transcriptional inhibitors. I think it's a great suggestion that we should do. I mean, from everything that we're seeing regarding downregulation of the transcriptional and translational machinery, we expect to see the same effects, but we haven't actually done the experiment. So the last question from Yi-Yung Lee. If you increase DNA content in a big cell, would it prevent the cells from being less prolific? Yes, I mean, we have, as I already answered these questions. Then you're asking me about the differentiation potential of X-large HSCs. So we have looked at this. I went over that quickly. Both in vitro and in vivo, we do not see differences in differentiation potential. Blood composition is not altered, but the contribution by the large HSCs is become much smaller. All right, and so I'll leave it at that. Thank you very much for all your questions and your interest. It was a real pleasure telling you about our lab's work. Thank you.
Thank you, Angelika. Thank you very much for this great lecture. And I thank also the audience for taking part in this and being with us today. We look, of course, forward to see you again next year, hopefully in person. And in the meantime, I invite you to visit the websites of both the Austrian Academy and ÖAW-IST Austria. You see the URLs on your screen, because we have many different lectures, especially in a year like this, that are now online and easily accessible to everybody who is interested. So also now, in the name of also Anton Zeilinger, let me wish you all the best. Please stay safe and healthy, and goodbye.